predictor today. I actually learned a lot myself going through all the various details of the various splice predictors and kind of marveled at how I can really get sort of blinders on and not really realize all the features in doing what I do every day, which, by the way, I need to disclaim that I am a full-time salaried employee of Ambry Genetics, and this is not so much a disclaimer, more so a plug, that I am the coordinator for this ClinGen HBOP group, which is hereditary breast, ovarian, and pancreatic cancer. And although we're not ready yet, probably we'll put out a desperate plea for biocuration help, because we do have 7 genes that we're going to be working on. So we plan on sort of simultaneously developing rules for other genes at the same time that we start doing biocurations for the first round of genes. And so it's going to culminate in a lot of effort. So in future, if you get a request from me, it's this group. I also do want to put in a plug for, this perhaps being sort of more of a conversation and a demonstration and less a bit of a presentation, because I think that you don't know what you don't know. So the more the merrier in terms of, what splice algorithms are out there. I'm only going to demonstrate 3, well, really 4, that I use on a regular basis in my work, and so I don't necessarily know what else is out there. So if you want to chime in, I would most welcome that. So I thought I would just sort of start with RNA basics, which is, perhaps just a little bit painful, but I don't want to spend too much time doing it. Let me just pull up one thing on my other screen. So I animated this because initially it looks a little bit overwhelming, but I think it is important to consider the RNA splicing basics here. And so everyone knows the dogma of DNA to RNA to protein, but that's really oversimplified, of course. We all know that. So I'm actually just going to introduce or add or remind us of 2 extra steps. One is that DNA actually doesn't just jump to mRNA. It goes through a pre-mRNA intermediary, and that's what's demonstrated here and is relevant to the spliceosome. And the other one that we'll talk about briefly is just nonsense-mediated decay in just a few minutes. And so, excuse me, the pre-mRNA contains all of the introns. So this is the RNA sequence, and what happens is certain snRNPs, so these are a combination of RNAs complexed with a whole host of proteins. And to simplify it, the ones that we know about, at least in mammalian systems, are U1, U2, U4, U5, U6. The U stands for uridine. These happen to be uridine-rich. And so U1 will bind to the splice acceptor site, and U2 actually binds to the branch site. And at that point, complex B forms. It brings in this complex of the human U4, U6, U5, also known as the tri-snRNP. And at some point, there's a rearrangement of this complex, and a couple of them leave. These get recycled to continue the process at the same time, to continue the process for other splice sites. And then the splicing actually occurs, leaving this intron to come out in the famous lariat shape. I don't know, when I was in school, they really thought that was important. I don't know why the lariat is so important, but so this is the piece that gets spliced out. And so then you have your mature mRNA. So the exons are retained, and then everything kind of gets recycled. So the key features here are the 5' splice site where U1 binds, the branch site, which is an adenine residue where U2 binds. Then there's a polypyrimidine tract that becomes important. There's a 3' splice site. Some of the components, by the way, of the spliceosome are actually left behind, and they form the exon junction complex. So this EJC forms a record of the splice events, and that becomes important for understanding NMD, and we will get to that. And so I think it's definitely useful to go over the consensus U2 splice site. So this is just a compilation of all the mammalian splice sites that are known and how conserved each residue is relative to its location in the intron and in the exon. So the height of the nucleotide refers to the degree of conservation among all the different splice sites that were analyzed. So the bigger that nucleotide is, the more highly conserved it is. And as becomes immediately obvious, this plus 1 and 2 and the minus 1 and 2 on the other side are invariable in a U2 splice site. And that's why we have the really strong weights in PBS1 for these canonical splice sites. And we'll look at PBS1 briefly in a few slides. One thing I do want to note is that the -4 position right here, this N, can be any nucleotide. And that basically means that, substitutions here are almost invariably tolerated. And we've definitely seen that in a lot of the functional studies that we're doing at Ambry, and I think obviously in the literature this is pretty well known too. Another thing to note is that the last nucleotide of the exon here is almost always a G residue, and the ACMG guidelines don't account for the fact that this particular residue is very highly conserved. We give it extra weight internally, and I think probably some other biocurators do as well. So even though that's not outlined in the ACMG rules, this last nucleotide is actually very important, and substitutions here are frequently not tolerated either, almost as much, although not quite to the same degree as these plus and minus 1 and 2s. Okay, and then another point that I wanted to make is that in general, there is a belief, and there's some empirical data, that the further away you get from the canonical splice site, the weaker the splice defect could be. So, an alteration at +1 might lead to a complete exon skipping event versus something at +5 might lead to the same exon skipping event, but maybe at only half the amount of what's occurring if you adulter the canonical splice sites at +1 and 2. So I did want to bring up U12 splice sites. Sometimes there's concern over these splice sites. What am I looking at? Sometimes people don't know if they're looking at an unconventional U2 splice site or a U12 splice site. I think it's safe to say you're looking at an unconventional U2 splice site. U12 splice sites are actually extremely rare. They're less than 1% of splice sites. They're sometimes referred to as an AT-AC site, and that's because this A right here, you can see this is not at all available for use in the U2 splice site. So an A is tolerated in the U12 type, and on the other end, similarly, this -1 has room for variation as well, either a G or a C. So sometimes they're referred to as an AT-AC. But obviously, quite frequently, they can also look like a U2 with a GT-AG. So that's the same as the U2, GT-AG. And so they are recognized by the U12 spliceosome, so that's a different subset of proteins. They lack this extra long pyrimidine tract. You can see here it's very long in the U2 type, but it's much shorter here in this U12 type. These are not predicted well by in silico splice models. So if you're looking at one, don't try to use the tools that I'm showing you to predict what's going to happen when you introduce a mutation. We recently came across an interesting paper that says these particular U12 sites seem to be enriched in specific genes, particularly those involved in information processing. So these would be like DNA replication, repair, transcription, RNA processing and translation, but they can also be found in genes related to cytoskeletal organization, vesicular transport, and very particularly voltage-gated ion channel activity. So if any of you guys are working on those flavors of genes, you might actually be looking at a U12, but I think they're just really quite rare. Okay. So everybody's favorite topic, nonsense-mediated decay. You can't really talk about splicing without briefly talking about nonsense-mediated decay. I'm a very visual person, and so I find this diagram instrumental in interpreting certain alterations because it's not always straightforward what you're looking at. And I have a couple of cautionary cautionary situations that you might want to consider when you're looking at certain splice variants. So again, I'm only talking about splice variants. There's a whole other set with frame shifts and nonsense mutations, et cetera. And so the bottom line is I told you previously that this exon junction complex is left sort of as like a history, like a recorded history of where the splice sites took place. And so they exist on the processed mRNA. And the ribosome kind of chugs along doing its job translating this mature mRNA, and it encounters an exon junction complex, and it just keeps on going. And as it goes, these kind of get bumped off. That's the basic kind of like kindergarten tale of what happens. And so the problem is when it encounters a stop codon that's premature, it can tell that it's premature because it kind of looks in front of it and says, there's an exon junction complex in front of me. Something's wrong. This is a premature stop codon. I'm sending this to be degraded. And so off it goes. And so one important concept is this NMD site. So this is around 50 to 55 nucleotides upstream of the donor site and the penultimate exon. And the reason that is, even though it's still in the penultimate exon, the reason is that the ribosome itself is very large, and as it approaches that NMD site, it actually obscures the EJC complex itself. And so if it encounters a stop codon there, it can't, and I'm making air quotes, but you can't see me, it can't see this EJC complex because it's essentially sitting on top of it. And so it does not, anything after that or around there, it doesn't interpret that as premature, even though it is. And so it will not send those to be degraded. Okay. So anybody, I'm going to pause for just a second because that was a lot of background. If anyone wants to ask a question or input some information.
This is Deb. I just want to comment for the nonsense-mediated decay. So that's kind of like the stereotypical, but when we've been looking at it for VHL and going into functional literature of testing nonsense-mediated decay, it's sometimes different, different lengths than kind of this, expected amount or something like that. So it's always make sure to look at functional papers for that information too.
So from a gene-specific standpoint.
Yeah,
Yeah, that's a good point. And I remember having a conversation per email with Jesse Messer at one point about a particular gene, patched 1, which, and this is probably just an aside and we should keep moving, but that one seems to be really strange in terms of NMD. It does have an exon in the 3' UTR, which kind of creates some exemptions for it, but it's definitely not always a straightforward tale. This is really basic.
Yeah.
Thanks for that input. Yeah, there are some gene-specific nuances for sure. Okay, so these are the ACMG rules that involve splicing. And again, I have a lot of animation because they tend to fill my slides and then they look frightening at first. So excuse me while I click through these. The most obvious one is PVS1, which considers all of these loss of function alterations, including the ones with the canonical splice sites of plus and minus 1 and 2. There's also well-established in vitro or in vivo functional studies. I'm not going to touch on this very much today, but this would be, this also includes any splicing functional studies. I will really briefly touch on quality for that and interpretation. There's also, this is the highlight of this talk, the multiple lines of computational evidence. So this encompasses both sort of protein astrogenesis, but also the splicing impact. And then, of course, BP7 all by itself, which are the synonymous alterations, and how to decide if they are predicted to impact the transcript, because they obviously don't impact the protein. So today we're gonna really focus on this rule here, but you can't ignore the other ones. And so what are the aberrant splicing impacts that you might encounter? So for canonical splice alterations, I think the majority, at least in our experience, and I think in the literature, is exon skipping events. These might be single exons, or they can be multi-cassette, meaning more than one exon involved at the same time. Typically, if the exons are quite small, I don't think anybody really knows why some create single exon versus multi-cassette events. But basically, when you're interpreting how this might affect the transcript or the protein, you could, for the most part, say if it's going to cause an exon skipping event, that splice sites that weaken or abolish that native site when there is no availability of a cryptic site nearby, the donor, it's going to cause skipping of the closest exon. And so donor alterations will cause the skipping of the preceding exon, the exon right before it, and then acceptor alterations will typically cause skipping of the following exon.
Now, I think that I think there's a lot more instances where the donor alteration causes non-splicing of the following intron, and it's undetectable in RT-PCR and so on because the unspliced product is so big that you don't see it. So you maybe end up in a situation where there's the mutation, some fraction of the transcripts are being affected. Of the affected transcripts, 80% of them are including the following exon, which you don't see because it gets destroyed by NMD and it's hard to measure. And 20 of them are exon skips that you can then observe and are left with the impression that the sequence variant is causing exon skipping, but that's only a fraction of what it actually does.
Yeah, that's a great point. So the data that are available out there are biased because of the limitations of the assays that detect them. So yeah, that's an excellent point. I agree. And I guess that the answer is we don't know. We know that this definitely happens, but to what degree over other anomalies, we don't know. Thank you. Other potential splicing impacts are partial exon skipping. So this would be like use of a cryptic site where you lose part of the exon and retain other parts of it. There are intron retention events, as Sean just mentioned. These are very hard to detect because the size of the introns sort of preclude the capacities of the polymerases that are used in the assays that detect them. And then partial intron retention events. This would be like use of cryptic sites within the intron. And then in terms of non-canonical splice alterations, the exonic ones that include synonymous alterations, so these would be ones that are within the exon but not necessarily near the splice site, could create a cryptic splice site, and they could also affect some ESEs and ESSs, which we'll talk about, I think, in a minute, just very, very briefly, or they could cause cryptic site use, and similarly with the deep intronic alterations. So these are also not very near the splice alterations, but they may cause partial intron retention events or cryptic exon use. I think one of the most fascinating, and other things entirely, so one of the most fascinating ones that I ever saw was, this is called exonization in some terminology, where a particular alteration, it wasn't a single nucleotide variant, I think it was a small deletion, mutated a sequence where a specific suppressor would bind, and the suppressor was responsible for suppressing this exonization event. And so it increased, although in a hypomorphic way, this exonization, which caused a frame shift and resulted, I think it was ornithine, OAT. I'm a cancer person, so I can't, ornithine something or other. Anyway, that was so all kinds of things are possible. Okay, so let's look at PVS1. I just have one slide. I've highlighted the PVS1 is essentially recapitulated among the different, all the concepts are recapitulated among the different types of alterations, but I've just taken this excerpt for the plus and minus 1,2 splice sites, specifically for U2 splice sites. And the way I look at it is it's split up into two steps. Step one, is this going to be degraded by NMD or is it not? And then after you've answered that question, that path, you have to basically ask yourself, in a situation where it's going to be degraded, is there a transcript that could rescue it? So like an alternative transcript that excludes that exon, right? So is it present in biologically relevant transcripts? And then for those that are not going to be degraded by NMD, how important is the affected region? And so you get different weights depending on how you answer those two questions. And the suggestion from the SVI and how to answer those questions is take a look at the biologically relevant transcripts, look to see if there's loss of function variants in the general population. If there's loads of them, that exon is unlikely to be important, biologically important or clinically important. And then how much of the protein is lost. If it's, but arbitrary, if it's greater than 10%, it's more likely to have an effect than if it's a smaller portion. So these are sort of the two cautions that I have when you're considering splice alterations and trying to make predictions, besides the valid point that Sean just brought up. Last donor alterations, I just don't think that there's any way to predict what these will result in, and the reason being, and depending, on which camp you're in or how you're looking at it, if the donor site is going to cause a skipping event, there's nothing to splice into on the other end, and so I don't know if there's any consensus on how these turn up. I've never done any research myself, but I suspect it's a range of different kinds of anomalies. And the other one is, and this is just, sometimes we get a lot of questions about this, what happens when the penultimate exon, where the NMD site is predicted to be gone or is shown to be gone. A lot of people just think, oh, well, if it causes a frame shift, it's going to be degraded by NMD, but that's not really the case, because the NMD site moves essentially to the new penultimate exon. Yep. So now this third from the end exon becomes the penultimate exon. And if you think about it visually, like I usually do in that diagram, you'll realize that this alteration is not going to undergo NMD. And so you really need to infer how important is this frame-shifted region. Okay, so this is the only slide I have on the functionally validated splicing alterations, which fall under the PS4 for the pathogenic side or the BS4 for the benign side. High-quality splicing data always trumps in silico predictions, and I'm going to show you in the next few slides how often these predictors are wrong. We did a colleague of mine did some testing on this. Very quickly, how to determine what is a high-quality splicing data. You want to look at whether or not patient material was used versus a mini gene. In my experience, mini genes are okay, but they're often wrong, or they only show you one splice event when two different splice events might actually be happening if you looked at the patient material. And then you want to know if your assay is a quantitative assay, a semi-quantitative assay, or maybe not so great at all. So the quantitative assays would be, for example, a clone-seq assay versus a colony sequencing assay. So these are very similar, except the colony sequencing assay really only looks at a handful of colonies, maybe 20 or so, versus a clone-seq assay is able to do sort of like a deep sequencing on the order of thousands of clones. And so you get a much more accurate picture of how much aberrant splicing you're looking at. RNA sequencing methodologies, SNP analysis is also instrumental. So if there's a single nucleotide polymorphism that's within range, you can see if there's any allele bias and then decide, how much of the aberrant transcript is being produced from the affected allele versus not. A lot of times in the literature, you'll see gel images where people kind of look at it and eyeball it and say, well, this band looks darker or brighter, and so I'm going to estimate that, it's 40% aberrant splicing or something. That's really, semi-quantitative is actually generous for a description for that. Some people also look at Sanger peak height. So if you Sanger your data from a non-clone, just Sanger your straight-up PCR, you'll get different peak heights depending on how much of each transcript is present. But again, that's definitely semi-quantitative.
The approach is really difficult unless you're looking at chromatograms from both strands, and unless you also have normal samples to compare to.
Right. And you also, the other caveat to this is that in a, PCR, certain amplicons are easier to amplify based on sizes, and so they could be artificially inflated just by virtue of their size. Okay, so as I mentioned, the in silico predictors have a relatively high rate of false positives and negatives. Just so you can think about what they mean. A false positive would be the program saying that there is a splice defect when in fact there isn't, and a false negative saying that there will be no impact on splicing when in fact there is. And just to note that we know the existence of these exonic splicing enhancers and the intronic splicing enhancers are incredibly important. So all those features I showed you in the splice site, the 5' splice site, the 3' splice site, the polypyrimidine tract, the branch site, those are all important, but they're not sufficient for splicing to take place. These elements need to be present and guide the spliceosome as well. Some algorithms predict them and predict changes in them, but I think, I would always take that with a grain of salt. I don't think we understand enough about that yet. So these are data compiled by my colleague Heather Zimmerman. She was testing 5 different Splice algorithms to see how they performed against a set of 62 internally validated splicing alterations. And she actually, this is an unfair slide, I only have one on it. She did an extensive amount of work testing the thresholds for these alterations to see how they performed best and how they combined best to create the best predictors. But I'm using this slide to basically highlight the vast number of false positives and false negatives. So in gray, there are the false positives. So splicing predictors said, hey, there's going to be a splice site here. We tested it, and actually everything looked fine. Everything was normal. False negatives are the really bad ones. We don't want false negatives, right, because those are unlikely to be ascertained. You know, if the splice programs say there's no splice effect when in fact there is, those are the pathogenic ones. We don't want to miss those. And so I'm going to look with you all at fruit fly, ESE Finder, HSF, and MaxEnt. Splice Scan 2 is not one that I'm going to go over in this presentation. I'm not very familiar with it at all. But basically, these were the best set thresholds. So this is a percent change of the splice site. So, you know, fruit fly performed best when there was a greater than 25% change in the score of the native versus the mutated site. And so you can see that it varies substantially, but consistently there's at least a 25% false positive and false negative rate. And so importantly, this is why there's only a supporting line of evidence given for this ACMG rule.
Hey, Marcy.
So,
sorry,
yeah.
Heather, I just want to point out one thing about
Thank
that
you.
last slide.
This is Heather Zimmerman, my colleague, who produced these data.
Yeah, I do want to point out that we were specifically testing variants that we were having trouble interpreting, so this does not contain very many plus, minus, 1, and 2 alterations. So the predictors do a much better job predicting a deleterious impact for the canonical changes.
Yeah, that's a good point.
You know, the plus minus 3, 4, 5, or the synonymous calls with predicting new cryptic sites, etc. So yeah, the predictors will do a much better job at the canonical sites than they do at the other sites.
Right, and thank you. And that actually reminds me, the other thing that I wanted to add is that the sample size again was 62, and so you know that's a relatively small sampling. But in addition to that, these weren't parsed in any way by basically abolishing a native versus creating a cryptic. And I just have this feeling that HSF will perform better for identifying a cryptic site, and that's something I'd like to look at in the future. And I think this also highlights the importance of using multiple predictors before making any conclusions.
Can I ask a question? This is Corin.
Sure.
So
what's
the recommendation based on the slide?
Can you speak up just a little bit?
Sure. What's the recommendation? Can you guys hear me now?
Sure, yeah.
All right. So what's the recommendation based on this study? Like use multiple predictors and average the score? What's the deal?
I mean, if I were going to ascribe a take-home message to these data, it would be, yes, definitely use multiple predictors and, you know, don't put too much weight into in silico predictors, I think.
Well, but, you know, the rule for the in silico predictors is supposed to be supporting. And
right.
Looking at these pie charts, you know, it's immediately obvious that everybody besides SS2 is going to reach a supporting threshold.
Mm-hmm.
They're all resulting in more than twofold, you know, better than 2-to-1 ratio of true positives to true negatives, and better than 2-to-1 of, I don't know how I want to say that firmly. They're doing better than 2-to-1 on both the pathogenic and the neutral side, so they're going to reach a supporting without.
So it's appropriate. Yeah. I agree. So I just quickly reviewed the other VSEPs rules to see how they're modifying or adapting the use of splice predictors. And so CDH1, T10, and TP53 each have a statement regarding the use of this rule. CDH1 is recommending at least 3 in silico splicing predictors needing to agree They prefer HSF and MaxEnt, but the BDGP and ESE Finder are also accepted. P10 seems to feel the same way. They didn't make any additional specifications. And
did they mention anything about the first intron of P10?
I didn't go into it that
the first
deeply.
Yeah,
Is anybody from P10 on?
Sean, we got your feedback previously, and we're good. Thank you very much.
Something special with that one.
Super special.
Okay. And then TP53 recommends the concordance of 2 predictors, and they specified that they would like everyone to use MaxEnt Scan and HSF. So there's not a huge amount of deviation, but different groups have their favorite program. Okay, so briefly on where to find your sequences. If you're not as fortunate as I am and have a database that just spits them out at you, you could use Ensemble to get to them. I was thinking of just showing everybody really quickly how to do that. I find Ensemble to be an amazing and very powerful tool, but it's not, I don't think, the most user-friendly thing. So this is basically the home page. I'm going to type in MSH2 in the top right search bar, and it's going to ask me what organism I want to look at. So I'm going to pick human is always the first one that comes up. I'm interested in human mutations. And then you would basically just pick your transcript here. These are by ENST numbers. Usually there's a REST-seq match, but I've just noticed, I don't know if anyone else has noticed this within the past week, these seem to have disappeared for a majority of genes. If anyone has insights into that, let me know. And so this is the part where it might be a little bit tricky, like where do I go from here? So I think the best spot to go is to select exons up here, and then you may need to configure this page. So I have my default set up to show me the entire intron, or you can, set it up to look at just pieces of the intron. And so if you go to configure this page, I clicked over here, you can select either show the full intron sequence, or if you just want to grab a certain number of base pairs on each side, you can change this to 50, click the check mark here, and it'll give you all of that information as you asked for it. And so you would just pull a piece of the exon and pull a piece of the intron and paste them into your program. You may also be able to get them from NCDI. I never have luck with that. And then MaxEnt and HSF will actually provide it for you as long as it's one of their curated genes. They have, I think, a substantial list, so most likely you're going to be in luck. So you might be able to just get it from there, and I'll show you that. How much do you want to look at? Generally, we look at plus and minus 50 surrounding the alteration. MaxEnt actually doesn't even use that much of it. You can provide it that information, but its algorithm does not even look that deeply around the alteration. You can also put a much larger sequence, but steel yourself, because the output is going to look very messy. It's going to be kind of confusing. It's a really good illustrator to remember that these ESEs and ESSs and ISEs are, still help point the spliceosome to the canonical site. So even though you might have a really strong cryptic, halfway down the exon, it's not necessarily likely that it'll be used because all of those other pieces that are usually around the splice site are not there. So I don't think it's always informative to look too far away from your region of interest. Okay, so that's just for reference. I did a demo on this. Okay, so this is sort of the next part of where we get into it. And my preference is to do this sort of by demo, but I made slides in case things are broken. I feel like HSF breaks all the time. So I'm going to swap my screen here for a second. I'm going to pull over you guys can look at my kids for a second while I pull over my stuff here. Okay. Does anybody want to jump in with anything before we start this part? Okay, so I did email the whole group some sample Splice variants, so nobody had to go hunting and pecking for any sequences. So I'm just going to pull and use the MSH2 one, which I think is the very first one I put on there. Yep. Okay, so it's this MSH2 C.1387 -2 A to G. So what I've input for you, if you're following along, is 50 nucleotides on either side of the alteration. And so there's a couple of features here that we can highlight. It's going to ask if you want to look at Drosophila or human. I think everyone is more interested in human on this call. So that's the default selection. And
I'm not sure if you're showing anything, but I still see your last slide.
Oh, that's interesting. Okay, let me just unshare for a second and make sure.
No, it looks fine to me. I can see it. Yeah, I can see it.
Okay, so we're looking at a website, right?
Yeah.
Yeah, it just suggested for me. I don't know what that was.
Okay, thanks for letting me know. Okay. Okay, so you can search either for the 5' or 3'. The default is set for both the acceptor and the donor. You can choose to include the reverse strand. I've actually never done that, and that scares me a little bit. So I've never done that. I don't know if anyone has found that useful in variant interpretation. And then the default scores are set to 0.4. And this is really the most primitive of all the three that I'm going to show you. So that's kind of it. And before I spit out the results, I'm going to just bring my slide back over because I want you to know how they've generated it. So these are the thresholds and matrices that they've used for the humans. And you can see that they, did a very thorough job of changing the thresholds and, based on basically they took a small subset of known splice sites and asked the algorithm to predict it based on the threshold that they provided. So of course at the top you have this really big score. So these are scored from 0 to 1. 1 is the best splice site that could be predicted. 0 is not there. And so when they did that for the 5' splice site, it identified 26 of the known splice sites, but the false positive rate was really low. So this is really bad, and this is really good. And so then they did a calculation for the correlation coefficient. And so as they drop that threshold, they're capturing more and more of the known splice sites, but their false positive rate is getting higher. And so the best thresholds are really in the middle with this highest correlation coefficient that captures the most number of known splice sites without identifying too many false positives. So I just highlighted in orange here about what they would be. A good splice threshold would be for the 5' and the 3', and they both end up being around, between 0.45, 0.5, something like that. And so I would note that when you look at the analogous tables for fruit fly, the best is 0.4. So I think that these are really set up for Drosophila. If you click over to Drosophila, these don't change at all. So be that as it may, my recommendation is to drop this down anyway, just to capture anything cryptic. So we can look at the difference here. So let's see. When we don't change the threshold, this is a native site, by the way, an acceptor site. Hopefully you appreciated that by the sequence title. It does a wonderful job of picking up this native site right here. So it scores 0.98. It really doesn't get too much better than that. It's a very strong native score. When you upset the -2 position, it's completely abolished. It can't score it. So abolished in this case means it's below your 0.40 score cutoff. There is a cryptic site that's available. It happens to be in frame. You can see where we might be going with this one. It scores 0.64, so that's like kind of medium. That's not terrible. It's not fantastic. So let's just change the thresholds really quick. We'll drop it by 10%. So we'll go to 0.04, and let's see if this changes anything. Okay, so it really doesn't change much of anything. It brings up a super bad donor site that doesn't even stress me out at all. So that's essentially the demo for this one. It's a very basic predictor. I just want to make sure I covered all my points that I'd wanted to. Oh, one other thing I was going to mention is that the start and end sequence. So the outputs are actually different for the acceptors and for the natives and the mutated. So you'll notice it gives you 4 kind of categories. This is the donor site for the native, any acceptor sites for the native, and then these are your mutated. So this is the donor site predictions for the mutated sequence, acceptor for the mutated sequence. And the output for the acceptors is actually 41 nucleotide sequence. That's the algorithm that they use to determine the acceptors. And then the donor is actually smaller. It's only 15 nucleotide sequences. And the start and end locations are relative to the total amount of sequence that you put in. So I highlighted that for us in the slides here, showing you that this is cryptic acceptor that's there, and you can see it starts at +3. So this is my input sequence. It's 1, 2, 3, and there it starts. So hopefully that will help orient you. Okay. So let's go to ESE Finder. this is the landing page. If you just, put ESE Finder in your search bar, it'll pull up, and this is the home page for it. The first thing you'll maybe notice is that this is actually not set up to detect splice sites. It's actually the default is for detecting splice enhancers. So you're going to have to change this first drop down to the splice sites tool. This predicts mouse ones as well, which I don't imagine are that different. It also predicts a branch site. I take all of these off because when you see the output with all of that information, it kind of gets overwhelming and it's not necessary. So I find the easiest thing to do is just to untick all of these things. And then you paste your sequence in here. So for both Fruit Fly and for ESE Finder, you have to use FASTA format. So you need this caret and you need this title. Otherwise, it won't work. You can just also input one sequence if you don't want to analyze a mutation. It will do that for you. I don't think Fruit Fly does that for you. I think Fruit Fly, you actually need the two. Okay, so this has a few more options than Fruit Fly. You can ask it to change the output. You can report only the best hit in each sequence. I definitely don't recommend that. You want to know about everything that's going on nearby. You can report all the scores in each sequence instead of just the hits above threshold. So instead of changing your thresholds, you might just click this box. I've never done that before. That seems like it might create a lot of potential splice sites. It's going to come out as like an HTML format, so it's going to come out, the default is set to show you a graph and some tables. If you want plain text for some reason, you can select that as an option. You can also email the results to yourself. I've never done that. I've never felt the need to do that, but the other thing you can do is you can, in this case, you don't have to paste a file. You can upload your file by choosing a file. All right, so let's look at the results. So I have not changed the thresholds here. And so this is the same sequence that we used before, and you'll notice that there's no cryptic site predicted right now. We saw a cryptic site in Fruit Fly. So this is a very strong score. I couldn't really find the range of scores for ESE. This program was built mostly as per the title for identifying ESEs and not splice sites. They kind of just threw the splice site stuff in there. So there's relatively little information on the details that, on how they derived the scores. And so what you can appreciate here is that this is the table for the native, and then if you go down here, it shows you your mutated sequence, and you can see that there are no results. So the alteration actually abolishes that native site. And remember, abolished is only relative to the threshold that you asked it to look at. So we asked it to look at 6.67, the default. I recommend, again, lowering these. I'm going to lower it again by 10%. We're at 0.6. And I'm going to ask it to look again. And this time it picked up a couple cryptic sites, including this same cryptic site that Fruit Fly picked up. So you can see how it scores 4.74810, excessive use of decimal places, but that's okay. And so that was previously below our threshold, which we had previously had set to 6.67. And so now we're picking up this cryptic site, which, by the way, is used. I can show you that later, the data for this alteration. So that's not a great score. It's a little bit below moderate, just based on my experience. If you're interested in the graphical score, I don't know why they picked these like really eye-popping colors. It's kind of hard to look at, but the 3' splice sites are pink and the 5' ones are red. And if you forgot to untick the boxes for mouse, those will show up here in different colors too. So I don't find it useful because they don't graph the mutated next to the native. So I generally ignore these altogether and just take a look at the scores. Let's make sure we covered everything on that before we move on to my favorite. Okay, yep. Let's go to HSF. Okay. So HSF is quite powerful. We have 15 minutes. I might kind of try to get through this as fast as possible, but basically you got to start an analysis or analyze now. This has a whole lot more features. I'll try to get through them one by one. Before we do that, I want to mention what HSF has set for thresholds. So HSF is actually, this website builds HSF and MaxEnt, which are two different algorithms but provided all on the same website. So It's a two for one. So HSF, the values range from 0 being a non-splice site score and 100 being the strongest score. The threshold is said to be set at 65, but you'll see later in the graphical that it actually looks to be at 70, but it calls lower scores than that. A good splice site is considered above 80. And for HSF, a broken splice site is defined as an above threshold site, so if it's not above threshold, it doesn't count. And the difference in the score has to be less than minus 10%. And so the cryptics will be opposite that, right? So a new splice site, which is use of a cryptic site, is defined as a below threshold site where the difference is greater than plus 10%. So for MaxEnt, the scores run from minus 20 to plus 20. Plus 20 is the best. I've never seen plus 20 or anything even really close to that. So I think that's kind of unusual. The threshold is set to 3, so anything scoring outside of that is not considered a splice site. A broken splice site is the same as I just described, but it's a 30% difference, and the same for cryptic. It's for, positive for 30%. All right, so let's go back to the program. There's many different options in each of these drop-down menus, and there are features that are associated with each that are good and bad. So if you want, I usually analyze a sequence. This is what I do, and I usually paste my own sequence because it's provided to me. So let's just start with that. That's the same splice site that we've been looking at. If you really want to, if you put in a very large amount of information here, you can highlight a certain part. Like if you only want to look at like 20 nucleotides around it, but you put in 50, you can add that information here. I don't really do that. It seems to take a lot of time. Here's one really important feature. Okay, so I'm analyzing a sequence, so I can only analyze one sequence, not two. So I'm going to put in the native site here, and hopefully you can appreciate that the capital letters are the exonic part, and the lowercase letters are the intronic part, and then I've highlighted the spot that's supposed to be mutated with a capital letter there. Do not use FASTA format. No carets, no title, just the sequence. It'll get messed up if you try to do more than that. You can also ask it to add certain matrices. So NN splices in here. The default does not include that, and I'm just going to keep it simple and not do that. You can also change the thresholds for MaxEnt. Never really done that. So there's some things in here you can ask it not to indicate the branch points, to ignore the ESE Finder stuff, to make it more simple. But, I think you can just do that on your own mentally when you look at the results. So let's proceed to the analysis. And you get a couple different ways that it's displayed. So this is your input sequence, and these are all the potential splice sites predicted there. And you can clearly see, and you might have to look at the pattern to make sure that this is the native splice acceptor site here. It scores 95, which is very strong. If you want to zoom in on any of these, you can click and drag down here. And if you want to get out of that, you just click outside the part that you dragged in. And as hopefully you appreciated, if you hover over it, it'll tell you what it is. It's a donor site, so purple circles are donor sites. It'll tell you the score. It'll tell you the start position relative to your input sequence. Blue squares are acceptors. So now I don't know why this, and sorry, the top part here is the HSF matrix, and the bottom part is MaxEnt. So they're totally different programs, but they are predicting some of the same splice sites. And as I mentioned before, that the yellow part is anything above 80, dark yellow is considered a strong splice site. And then even though the threshold is set to 65, it's giving you a light yellow highlight for like an okay splice site, which is above 70. So in order to get the tabular data, you actually have to click on this ribbon down here to ask it to look at them. The first tab is potential splice sites. The others are for branch points, enhancers, silencers, and other splicing motifs. I don't really look at these, again, because I don't think we know how to interpret them yet. And for whatever reason, it defaultly opens HSF. You have to ask it to open MaxEnt as well. And so these are just the tabular form of the graphical depiction here. So now if you want to ask it to look at a specific mutation, you can ask it to perform a quick mutation. It's going to say, where do you want this mutation to be? And I want this mutation to be at position 49. That's the location right here. And I want it to be, what was it, a G. I don't want it to be an A anymore. I want it to be a G. So I'm going to proceed to this analysis. But before I do, I'll show you, you can also perform deletions, insertions, and indels. You just have to fill these boxes in correctly. every once in a while it takes me like 2 or 3 tries to get it right because it's a little bit hard to count where you want it to start. But fortunately, it shows you exactly the change that you made. So it's highlighting in green and red what the alteration is. It gives you
Marcy, do you mind
yeah.
if this is Jonathan? I just want to make a quick editorial comment. I think, you know, your point about having to manually enter things is exactly why we need informatics support to do these types of analyses, right? Computers can be taught how to enter things into the appropriate boxes so that you don't have to do it and make mistakes doing it. That's an editorial comment. I know that we've got lots of development needs, but it seems like if we really want to be able to use Splice site predictors, that we need to figure out a way to make this something that curators can use without having to manually enter.
Yeah, there's definitely a lot of room where you can introduce error. Yeah, and, you know, certain people may have ways to avoid that based on, you know, internal algorithms that give you the sequences.
But I think the way to handle it is to decide on an algorithm or two, and then run all possible single nucleotide substitutions through the algorithm and record the results.
Just pre-filled.
Yeah, I mean, that's what we did for BRCA1 and BRCA2. It's just sitting there in the priors database and sitting there at UCSC.
I know groups, I don't want to derail this, but I mean, a lot of the expert panels have used professional programs like Alamute, for instance. They haven't manually done this. They've relied on those programs to run these splicing predictors for them.
Mm-hmm.
I mean, it's great to find out how to do it manually because, like, I don't have access to Alamute, so I want to know that, first of all, how they work. But I think just in general, where we're actually getting our information on splicing from for expert panels, as far as I'm aware, is someone at a laboratory has access to Alamute and has been running it.
Right. And right now, I mean, the current recommendations for the VSEPs that are, you know, accepted are asking you to use these programs. So we do need to know how to do them until something better comes along. Okay, running out of time, so I just wanted to make a few points here. And one is that when you do perform a quick mutation, you only are seeing the results for splice sites that changed. So you'll notice that the cryptic splice acceptor site of interest is not in this table. And the reason is because it didn't change in its score. It's a valid, relevant, alternate splice site that, as I mentioned, is used. But when you do this, it disappears from the results. So be careful. Don't forget to interpret that. There's 2 other ways that you can look at this. One is if you look at a mutation instead of looking at a sequence. This is where you might have less room for, like, where you might not create any errors. So as long as you have the right input sequence, this is the alternate sequence with the G substituted there. And this might be something some people don't know. In this mode, you can ask for a graphical representation. So previously, it wouldn't give you the graphical. If you click those advanced parameters and you ask it to add images, it will give you the graphical image of how those scores are changing. So you have to ask it to open that, and you can see in red and green how this native splice site drops below the threshold. But again, you're only seeing the changes. So if this were the only analysis that you did, you would miss that alternate, the alternate cryptic acceptor site, and you would misinterpret these data. So just use that feature with caution. And then one other thing I wanted to highlight here is that you can analyze a sequence by gene name, and you've got to make sure you're looking at the right transcript, and it'll ask you to. So let's look at MSH2, and let's look at exon 2. It's not exon 2 that we've been looking at, but and it's going to ask me which of these transcripts do you want to look at. They've only got a few of them in here of the many that are in Ensemble, and it's going to ask me how much of the surrounding exon or intron do I want to look at. The default is 100. Okay, that's great. So now it's going to give me a nice graphical representation. I can still perform a quick mutation. It's going to take me longer to count to where I need to be. But this graph has a few extra features. The white box and circle are the known splice alterations. You can still zoom in here, so if I was only interested in this acceptor, I could do that. Again, this is not the same splice site, so don't be confused that there's no alternate here. And then tabular format as well. Okay. I had a few, and I guess, hopefully that was demonstrative enough. I'm just going to run through the interpretation part of this very quickly. So the things you need to consider is, depending on whether your native site is abolished or decreased, you have to ask yourself, how much is it decreased? Is it significant? That is not those kinds of details are not provided in the ACMG guidelines. In terms of cryptic sites being created or strengthened, you have to ask, is it stronger than the native? Yes, maybe, but was it already stronger than the native? If it was, there's probably not going to be an effect. I would interpret that with caution. And then do the majority of predictors agree? So just so you know, this is the set of splice sites that we've been working on, the summary table. Everything agrees that it destroys or significantly impacts the native splice site. There's also a predicted cryptic site. And the conclusion that I would draw is that the native site is abolished, a cryptic site is identified, but it's not ultra strong. And so the reality is we have tested this alteration in-house. It does utilize this cryptic acceptor to a very strong degree. And this causes an in-frame addition of 9 amino acids because of 27 nucleotides that were encoded in the intron that weren't there before. The question was whether or not it was pathogenic. This did show up in a homozygous state in a patient without CMMRD. This is not pathogenic. So the take-home message is, please don't forget to lower your threshold and pay attention to those cryptics. Even if they're not super strong, like this is not super strong, this is not super strong, they might actually be used. This was an interesting synonymous case. There were absolutely no changes predicted at all. So we would throw this one out the window. In reality, this alteration is detected repeatedly in patients with very classic von Hippel-Lindau, PCC, PGLs, and hemangioblastomas, and it was shown to cause skipping of exon 2. So the take-home message here is in silico is a very small piece of the puzzle. Pay attention to the phenotype of your patient, look at everything. It might be synonymous, but there could still be a story there. This is a trick question. This is a BRCA1 alternative splice site, so please don't forget to consider PVS1 and all the rules that it carries with it. So this abolishes that native splice acceptor site, but this particular exon is absent in a major predominant alternative isoform that appears to be functional. And the last one is, this is one that was outside of a cryptic site, outside of the very consensus site. And what it did was it created a very strong, these are fantastic scores, a very strong cryptic site. And it was used, and this particular alteration created a frame shift, and it was shown in an independent functional assay that can detect both protein and transcript changes, and it was not tolerated. So the cells all died, meaning it was not tolerated. So the take-home message is that cryptic site generation use may be rare, but it's real, and take it seriously. Okay. We're right at the end, so I don't know if we even have time for questions and comments, but I think people made some really great comments and had some good questions during the talk too, so I appreciate that. Thank you to all who contributed.
Thank you
so much, Marcy. That was really fantastic. We are at the top of the hour, but would it be okay if folks contact you if they have any questions?
Sure, absolutely.